Good afternoon to everyone, and thank you for participating in this webinar. Today we will introduce to you the EFSA guidance on allergenicity assessment of GM plants. My name is Antonio Fernandez. I'm Scientific Officer at the European Food Safety Authority. I'm here together with Fritz.
Yes, good afternoon. My name is Fritz Koning. I'm a scientist and immunologist. I work at the Leiden University Medical Center in the Netherlands. And I'm basically an expert on celiac disease, and that is why I'm here today.
Yeah, so we will both introduce to you this document, but before doing that, I will very quickly go through initial slides related to the guide to the attendees. So this webinar is being recorded. Please ensure that you have carefully read the EFSA disclaimer and data protection note. If you do not agree with the EFSA policy on webinars, please disconnect now. The webinar is in English, and the questions should be submitted in English as well. Volume and speakers, you are automatically connected to the audio broadcast, one-way audio, so you will only be able to listen. Check the audio panel control for adjusting your volume, and ensure that you are enable sound on your computer and turn on your headphones. The virtual room, in the slide, you will have what you see on your screen. You have the presentation window with the PowerPoint presentation showing. On your right, the Q&A box, where you will submit your questions to the content of the presentation. At the bottom, a video window where Fritz and myself will be shown. A chat box for technical issues that you have, and finally the small files box with the main links to the event. The questions that you will submit should be concise and submitted once. Please do not reiterate the question, and follow-up questions should be self-explanatory. Important to say, you can ask questions until 3:10, then it will be a stop and you will not be able to submit any more questions. You will see the answer right below the questions row once replied by EFSA. And the webinar is foreseen to last until 3:30. So your questions will be answered until that time, and then we will stop. If we are not able, together with EFSA colleagues, to answer all your questions, then please send those to EFSA App Desk, and we will answer those at due time. We have registered more than 100 people to the event, and these encompass different continents. So we have there the US, Canada, China, Taiwan, for example, as well as many countries from Europe. I will move now into the allergenicity guidance per se. A document was adopted in May 2017, this year, and it's mainly addressing three topics for allergenicity assessment: non-IgE-mediated adverse immune reactions, in vitro protein digestibility, and endogenous allergenicity. The first two topics deals with the assessment of the novel protein, while endogenous allergenicity is tackling unintended effects. Starting from the origin of this mandate, among the different institutions that can task EFSA, in this particular case is an answer to an EFSA self-mandate to consider new developments in the area, to address member states, NGOs, European Parliament applicants' comments, and to assist on the practical implementation of regulatory requirements, which are mainly Regulation 503. An important aspect we took into consideration was the stakeholder and public engagement, and big effort was employed in that side. We created a focus group, consultative body, engaging with all the different stakeholders, a very stimulating initiative. We organized a workshop at the initial stage of the guidance development, public consultation, and an info session as a step. From the technical aspect, immune-mediated adverse reactions to protein is a key element considered in the risk assessment by the different bodies. And taking Codex as the umbrella of all this, is mainly focused on IgE-mediated immune adverse reactions. in the new guidance, what we have done is going beyond that and taking the EFSA report, external report from the University of Manchester, consider different conditions, combine IgE and non-IgE-mediated adverse reactions, or per se non-IgE, and establish that for celiac disease, there is a clear cause-effect relationship, and for which we can define risk assessment considerations further. In the guidance document, you will have, we presented to you a stepwise approach proposal for the risk assessment, initiating with the knowledge on the protein as the first step. The knowledge on the protein should serve to calibrate what other steps are needed, studies, conditions for the risk assessment. If the information available on the knowledge of the protein is insufficient to support its safety, then a search for sequence identity is recommended. Fritz later on will further give some examples from this side. From the step 1, if no concerns are identified, then the risk assessment can stop at this moment. On the other hand, if concerns are raised in a step 2, we propose HLA-DQ peptide modeling, which again Fritz will provide some examples afterwards. If no concerns are identified, then risk assessment stops. If concerns are raised in a step 3, more elaborated in vitro testing are proposed. HLA-DQ binding assays, T-cell testing, or in vitro digestibility are those in the document. So as you have here, overall stepwise approach, risk assessment for celiac disease is, we can say, a comprehensive risk assessment approach put on paper for the very first time. I will move forward into the second topic, in vitro digestion. In this case, in the guidance, at the moment, at this stage, there are no recommendations in the form of guidance. However, EFSA proposed a refined in vitro digestion test and the need to have an interim phase to test the added value of this refined in vitro test. EFSA launched a procurement call in July. We are at the moment the selection step, where experimental data will be collected, where these tests will be challenged. Annex B in the guidance document contains additional considerations for the interim phase and examples provided. I have included here a screenshot of one of the figures in the document, where gastrointestinal conditions are suggested to consider different segments of the population. And in according to this, at least 2 gastric conditions are to be provided. I will not get into the details. You have this figure in the guidance. And as I mentioned before, this will be the starting point of the EFSA procurement to develop experimental data. Based on those experimental data, EFSA will further assess what is the added value of this proposal, and if it indeed adds value, what are the next steps for the final implementation in the form of guidance. And from this moment until that happens, EFSA will continue to base its assessment on the EFSA guidance 2011. Finally, endogenous allergenicity, focusing on unintended effects, as I said, and a comparative approach is suggested by international documents, Codex Alimentarius. It's in line with this approach, a substantial equivalence concept that was transmitted into the EFSA scientific opinions. So the paradigm is still kept in the EFSA guidance, the overall assessment. The analysis is to be performed on a case-by-case basis. In the case of Europe, is according to regulation for common allergenic foods, and to date, soybean is the main crop that is being analyzed. In the guidance, we also stress the possibility other plants that soybean could be submitted to such analysis whenever considered necessary. And for this, we stress the relevance of having risk managers, risk assessors, stakeholders, and health professionals working together in that view. Focusing now, what are the relevant allergens for quantification? In Regulation 503, European Commission further afterwards suggested to start the safety assessment focusing on the Table 20 of OECD consensus document. In the EFSA guidance, we propose to have an evidence check performed where all these different allergens included in this Table 20 as potential allergens are evaluated, taking into consideration the literature available, and additionally complemented with information in several databases, allergen databases available. Alternatively, we also suggest the possibility of having systematic reviews performed, where afterwards the applicant will have to discuss what are the clinical relevance of the different allergens. In the guidance, we provide an example in Annex 3C, how this could be performed. You have here a screenshot of OECD Table 20, a list of potential soybean allergens. In this example in Annex C, we highlighted those that are already included in international recognized databases such as WHO IUIS. And as you can see, Glyme7, for example, was not in Table 20, but it is included in WHO as a relevant allergen. In orange, I have included here those allergens for which there is a considerable peer review literature available. They comply with the criteria of WHO, and several scientists have suggested them as a relevant allergens, already publicly available. Finally, we have those in gray for which there is limited, not existent peer-reviewed literature available. And therefore, their clinical relevance is very weak at the moment. The relevant allergens should be measured. We propose quantitative ELISA or mass spectrometry to measure these allergens. And we also stress the benefits it will have to have a harmonization between the applicants with the view of a future development of an allergen compositional database. This will be used to consider natural variability in a more robust way. Historically, this assessment has been performed with the use of human sera, and the assessment was performed GM versus non-GM by visual observation, which, as you can see, these have the new recommendations has clear advantages. Finally, for the data interpretation, natural variability of allergens, as I was mentioned, is the starting point in the assessment and considered very important. EFSA developed a design of field trials where commercial varieties are included for these purposes to consider already at that stage natural variability. And on a case-by-case basis, of course, if the levels of expression goes beyond the natural variability, additional requirements will be requested, and this will depend on the magnitude and number of the changes, the clinical relevance of the allergens involved, exposure considerations, and eventually, if needed, clinical evaluation. Double-blind placebo-controlled challenges comparison GM versus non-GM, and dose distribution curves to single allergens will also be informative information in such respect. So with this, thank you very much, and I pass the floor to Fritz.
Thank you very much, Antonio. I will now go a little bit further into the details of the celiac disease proposal, or at least the guideline that we put out. And the first figure is really very important to understand what the basis of the guideline is. The first figure shows a very simplistic view of what is happening in the intestine of a patient with celiac disease. There are CD4 T cells present in the intestine of a patient with celiac disease. They carry a T cell receptor, and this T cell receptor is specific for a gluten peptide that is bound to HLA-DQ2 or DQ8, and this molecule is present on antigen-presenting cells, and these are also present in the intestine. So if this happens, you basically get tissue damage. That is the key feature of celiac disease. This is very well established. All patients carry either HLA-DQ2 or DQ8. Gluten is the environmental protein antigen that causes this disease, and these CD4 T cells are present in patients and not in healthy controls. So this is a key issue, and what makes celiac disease so special, we know the identity of those gluten fragments that cause celiac disease. And that is not just one fragment, there is a whole bunch of them. And this is a list that was published in a review a couple of years ago by me and my colleagues, and it lists the gluten peptides, the gliadin peptides, the glutenin peptides that are involved in celiac disease. So we have these sequences, we know exactly that patients can respond to these sequences. So what you can do in first instance is to search for 100% match with the T cell stimulatory epitope. So basically your protein of interest. You simply look if there are fragments in there that have 100% match with a gluten epitope. If this is the case, there is a clear hazard. Most likely, in that particular case, you are dealing with a gluten protein, because gluten is a very unique protein with unique properties. So if you have 100% match, most likely you have a gluten protein at hand. In other words, many proteins don't look like gluten proteins. But there is, of course, the possibility that there is a concern because your protein contains a sequence that has a partial match with a T cell stimulatory epitope. So now, how are you going to look if this epitope is a hazard, yes or no? For that, we looked in the table and we found that all these peptides have this very characteristic sequence that is exemplified by QLPY and variants thereof, which are listed over here. ELPY, QLPY. Very critical is that in this first position, there is always an E or a Q, and here is only a P possible. And then at this position, there are 5 possible amino acids, and here there are also 5. So you can basically make an algorithm that consists out of Q/E, then an X1, these 5 amino, either of these 5, a proline, and an X2, which is either of these 5 amino acids. That is a very simple search algorithm that you can use to look if your protein of interest contains this particular sequence, and that will focus you on sequences that potentially have some sequence similarity with gluten. Now, on top of that, we have very detailed, I forgot this. There are also some gluten peptides that don't contain the algorithm, they don't contain a motif, and therefore, in this particular case, you need some additional strategies to see if your protein could match these sequences, and I will further detail how you can do that just a little bit later. So on top of this, we have very detailed information on how these gluten-specific T cell receptors, which are in crystal structures over here, recognize the gluten peptide that is somewhere in the middle over here, which is bound to the HLA molecule, which is at the bottom here. These crystal structures have all been published a couple of years ago in a collaboration that we had with a group in Australia. There is very detailed knowledge in these structures, and this is an example of how a T cell receptor interacts with the gluten peptide in yellow and blue, which is bound to the HLA molecule, which is in gray, and the T cell receptor is this multicolored structure over here that is interacting with the gluten peptide. And what you can see is that there are critical amino acids that bind the gluten peptide to HLA-DQ8 in this particular case, so a DQ2, so there's one over here, there's one over there, and there's a third over there. So these are going down into the HLA molecule. They cannot be seen by the T cell receptor, but these residues over here, they are actually seen by the T cell receptor. And this is the most famous example. This is an arginine coming down from the T cell receptor. It interacts with the gluten peptide at multiple sites. And in addition to that, there is an interaction there, there's an interaction there, and there's an interaction there. So we have knowledge at the atomic level which amino acids in the gluten peptide are important for binding to HLA-DQ2 and which amino acids are important for binding to the T-cell receptor. And that is very unique. There is no other disease where we have such detailed knowledge. So you can use all this information to devise your search strategy. I'm just going to give you a few examples, some of which you can also find in the guidance. And I will explain a little bit further how you can then see if a particular peptide represents a potential hazard, yes or no. So the first example is when the motif is present. So the top sequence over here is a known gluten sequence. It's a very immunodominant gluten sequence, and this is a hypothetical sequence. And you can see that these two sequences have the motif, QLPY, in this particular case it's QLPA, and there are 4 unique or shared amino acids between these two peptides. So is this peptide now a potential hazard, yes or no? That is the big question that is out there. Now you have to realize that this proline to alanine substitution over here is pointing downwards and therefore is not seen by the T-cell receptor and does not really affect binding to HLA-DQ2. We know this. The same goes for this substitution over there. So if you look at this, we find that there are 4 identical amino acids, and these are critical for T-cell recognition. Here is a conservative substitution, and 2 of these substitutions are invisible to the T-cell receptor. In other words, you have a potential hazard. This peptide could trigger a gluten-specific T-cell. So you have to do more investigation to determine if this sequence is toxic or potentially hazardous, yes or no. Second example, another gluten peptide, very similar to this peptide, but in a slightly different frame. And this again is a hypothetical sequence. You will see that again, there are 4 shared amino acids. This is the QLPY, QLPA motif again. And then you have to realize that the substitutions in this particular peptide are much more likely to cause the T cell not to react. This tyrosine over here is incredibly important for T cell recognition in this particular sequence. So the substitution to an alanine is known to abrogate T cell recognition. In addition, the proline to serine substitution is also known to abrogate T cell recognition, and the Q to R substitution at the last position over here is known to inhibit binding to HLA-DQ2. In other words, there are a number of substitutions in this peptide that prohibit recognition, therefore there is no hazard. So this is when the motif is present. I'm going to give you 2 examples when the motif is not present. This is all based on an HLA-DQ8 epitope. This is a very immunodominant epitope in the gliadins, again, one of the groups of gluten molecules, and this is a hypothetical sequence. So now you see that there are 5 amino acids shared. And then the question is this now a potential hazard or not? You have to realize that there are 5 identical amino acids. 1 substitution, again, is sort of conservative, the phenylalanine to isoleucine. This is both are very large amino acids. But most importantly, the Q to E substitution actually enhances binding to HLA-DQ8. In other words, over here, again, we have a potential hazard, and further investigations would be needed to clarify if this peptide could be dangerous for patients. The final example, similar HLA-DQ8 epitope, another hypothetical gluten peptide. Actually, in this particular sequence, you can see that there are 6 identical amino acids. So there's a very high sequence similarity. But there are a number of critical T-cell receptor contact residues that have been affected. This Q over here and this Q is essential for T-cell recognition. And it's been substituted by a serine here, an alanine there, and therefore you have 2 major changes because these are 2 very small amino acids and these are very large amino acids. Therefore, there is no hazard. In addition to this, if you have identified a peptide that might trigger a T cell, you are therefore required to do some additional experiments. One of those could be peptide binding assays. You can determine if a peptide has the capacity to bind to HLA-DQ2 or DQ8. These are assays that have been published. There are several laboratories in the worldwide that have this assay operational. It is something that can be done. And this is one method we propose could be used if you have a very interesting protein that you want to pursue, despite the fact that there is a sequence that may be deleterious to a patient with celiac disease. If you can show that the peptide doesn't bind, then there is no problem. Likewise, you can do molecular modeling. This is actually a peptide that we modeled more than 10 years ago. This is a peptide bound to HLA-DQ2. This is a gluten lookalike that we were interested in. We modeled the peptide. You can then take the peptide out of the structure and look at the side view. And in the side view, you see which amino acids point down and therefore anchor the peptide into HLA-DQ2. And you see which amino acids point up and are potentially recognized by the T cell. And then you can compare the two sequences. This is the gluten sequence, and then we have the lookalike sequence, put them side by side, and you will see that where in the gluten sequence there is something very small over here, there's something very huge over here in the lookalike peptide that will prohibit the T cell from interacting. And likewise, there is a similar change over here, which will also inhibit T cell recognition. In other words, my conclusion is that potential endogenicity can be predicted, and that is the basis for the guideline that we put out. I thank you very much for your attention, and if there are any further questions, we are very happy to answer those.
Yeah.
Thank you.
Yeah. So we designated some time to respond to questions orally. So if you have any question, please go ahead. At the moment, we don't have any. So we wait a little bit, a couple of minutes. Hopefully some of you will kick off. A question that was popping up from previous meetings was related to the transition period that we didn't address here in the slides. It's more from the administrative side. We included transition periods in the guidance depending on the topic. For celiac disease, we included a transition period of 6 months that finished this month in December. So from now on, whenever we receive an application, this should contain an assessment related to the celiac disease. For endogenous allergenicity, we split it in two. If there is material available for measuring allergens, then we included 12 months, while if there is no materials out there, 24 months. For in vitro digestion, as explained before, we have no guidance at the moment, so there is no transition period.
So there's a question from Esmeralda Posada. Will the assessment for celiac risk apply to any protein or only to proteins coming from cereal? The answer to that is, in principle, it applies to any protein because, of course, the cereal proteins would by definition be a hazard. And it's really about the other proteins that we don't know anything about, whether or not those will cause a problem for patients with celiac disease. So any protein that will be put into a GMO should be scrutinized, in principle, unless you can make the case that this protein is already readily consumed by people and has never shown any problems.
In the new version of the guidance, we stress the starting point, the knowledge on the protein, as Fritz was mentioning before, and therefore that should be the starting point to calibrate additional considerations. We are reading a new question that is
from Laurent Buff, but we don't see the whole question, do we?
We cannot see the whole question. It's cut. It starts for the sequence identity search. We should look at the DQ2, DQ8 epitopes listed in Table A of the guidance.
And the motif only. No, that's not correct. In the slides, I explained that you can also have lookalike peptides that don't contain the motif, and that concerns the gluten epitopes that don't contain the motif. Those are the a few epitopes that have that property that they don't have the motif. In that particular case, you have to apply the alternative strategy that I talked about in example number 3 and number 4, when the motif is not present.
Yeah. There is another question also we received in previous meetings. What examples are there already? It's from Hayes Kletter of non-gluten molecules interacting with HLA and T-cell receptors. Yeah, we had this question before.
Yeah, we had this question before. It's a good question. We are currently working on a project together with a group in Australia to identify such peptides. And although it's not been published, I can tell you that there are peptides present in naturally occurring organisms that are present in proteins occurring in naturally occurring organisms that can indeed cause gluten-specific T cells to respond. So the scenario that we put forward is live. There are such peptides.
And as we said at the beginning, the principles, we try to match it, to align it as much as possible with IgE safety assessment, starting with the knowledge of the protein as the starting point in the risk assessment. So we have no more additional questions at the moment. Before we stop, just to say a couple of words of this document, which we consider a step forward for the risk assessment of allergenicity, which probably goes a little bit beyond that. Okay, we have a new question coming in. What is the rationale for requesting characterization of digestion fragments of more than 9 amino acids in length, given that bioinformatic analysis performed on 8 amino acid fragments already provide confirmation of homology? So I don't know if this question refers to
the celiac or to,
to IgE or to non-IgE.
Yes.
In the case of IgE, for EFSA in Europe, in the guidance, we don't recommend to use 8 amino acid matches approach anymore, but to perform the approach, the 35% amino acid match over 80 amino acids, 35% similarity over 80 amino acids. So if there are no match there, for cross-reactivity, of course, no issues are expected, but there are other aspects to be considered, not only cross-reactivity. At the moment, the pepsin resistant test is the test that is currently requested for the hazard identification. And this goes along the lines for the in vitro digestion test to substitute or to improve the information release from this in vitro digestion. So it goes beyond cross-reactivity.
And if it concerns the celiac epitopes, there is very overwhelming evidence that the minimal size of a peptide to bind to HLA DQ2 or DQ8 is really 9 amino acids. So that is why the 9 amino acid sequences are shown in the table. This is commonly accepted basically in the field.
So well, because yes, the last words before we finish, I was saying about the beauty of this document, starting with non-IgE, with celiac disease, where for the first time we identified a concrete proposal for risk assessment. For in vitro digestion, we go beyond the pepsin resistant test, as we said, and this hopefully in the future will have new insights on how to look at the assessment to understand what is the fate of the protein in the gastrointestinal tract, which will also have new views for the hazard identification step, I'm pretty sure. And finally, for the last, for endogenous allergenicity, and again, if data comparability is allowed, hopefully in the future, an allergen database will be very, very useful. And not only for GMOs, but this again will go beyond that. And finally, for public benefits. So this will be a win-win situation for everyone.
I agree completely. Yes.
I think there are no more questions there. There are now 52 participants still there, so thanks a lot to all of you for this. And that's it.
If there are any further questions, I'm sure you will find us. Thank you very much.